Ready? Good morning, everyone. Welcome to San Francisco. Welcome to SWOG. Welcome to spring. Welcome to the Oishi Symposium. We're all glad to be here. It's good to see you. Good to see old friends, new friends, and here we go. It's going to be a great day today. We're here today at the Oishi Symposium, and for those of you who never had the opportunity to meet Dr. Oishi or his wife, Jeri, this symposium is named after them. Dr. Oishi was a lifelong investigator with SWOG and a PI from Hawaii. His wife, Jeri, was a research nurse, and they took good care of our CRP, ORP, RN, research nurse, CRA, all of our groups. And this symposium for years was sponsored by them, and certainly we want to honor them and thank them for their long time support for us. This is the ORP committee and subcommittee org chart, just so you know who we are, where we are, but we're your committee. There are 10,000 people now at SWOG that are in this category of ORP, oncology research professionals. So you're not all alone in the field, although sometimes probably feels that way. There's lots of people that work together hard to make sure that we can bring our studies to our patients at all of our sites. I want to acknowledge our executive committee members. This is the list of names on the executive committee. We want to especially acknowledge two of our longtime lifelong members, Annette Betley and Seal Petrovsky, who both just recently retired and aren't here with us today. So some of us were caught by surprise by those retirements. If you know them and want to reach out to them and say, hi, we missed you, that'd be great. We also want to welcome Erin Sebula and Liz Edwards to the executive committee. If you'd like to be more deeply involved behind the scenes, there are ways to do that. You just go to the members resources on the website, look for membership, and there's a link that will take you right to us and tell us what you'd like to get involved in. We always have openings and places to plug in. Although there are no formal CE credits for this meeting, you can print a copy of the agenda, or you can take home the agenda that our lovely Christine and Amy and others handed out at the door. And at the bottom, you can fill in how long you attended and keep that in your record for your CE tracking for SOCRA or ACRP or whatever you may need. So without further ado, we're not going to do a lot of announcements. We're going to get right to the heart of today's beautiful symposium, which will be a lot of fun and very interesting. And I'm going to turn this over to one of the co-chairs of our education committee, Joyce Tall. She and Deb Bergevin have worked really hard in leading the education committee to put today's offerings together, both this symposium as well as the open forum afterward. We hope you have fun, enjoy us, and learn some new things. Joyce?
Thanks, Connie, very much. And thanks to all of you for attending today. Deb, do me a favor, stand up for a minute. This is my co-chair, and she and I have worked with a team of tremendously dedicated folks in the education committee, and they have put all this together today. They're very talented. They're extremely energetic. They think about all new things all the time. And the most important thing is they truly come up with novel and unique ideas. So this year they came up with a really interesting idea. Wouldn't it be neat to showcase all the things that go on in the background by a number of people who are faceless to many of us, but really help to get a protocol out the door? Well, then it expanded, and somebody said, I know an investigator who just provided a talk about this, and that was Maggie, so from Fred Hutch. And then it expanded to, why don't we ask him to come? And then one of our very own was kind enough to step up and say, I'm a survivor, I'll talk about that. And I survived a clinical trial, and I'm working in cancer research now, which is pretty cool. And it became this team effort. Well, then it looked for a theme. And as you can see, this spring, the ORP Education Committee decided to take you on a very long journey to a place where there wasn't any trouble. Not a place you can get to by a boat or a train. It's far, far away, behind the moon, beyond the rain. And it's where happy little bluebirds fly and dreams actually come true. Now, if any of you were around last night, you listened to a survivor. And Toby was just phenomenal, because Toby discussed what it was like to do a trial. And that really capsulized what we were trying to bring to you this year. To do this takes a lot of guts, determination, resilience, courage, and working as a team. And so we couldn't think of a film that showcased that better than The Wizard of Oz. So we're bringing you along on our journey this year. Okay. We have a number of speakers. They come from as diverse group as the NCI to the Stats Center to operations to data. All aspects of this are going to be represented to you. So it's not just an opportunity to hear what goes on behind the scenes and appreciate the vast machinery that puts this all together, but it is also a tremendous opportunity to get to know some of the faces. If you see them afterwards, go up and tell them thank you, because many of these people do this job for years and years. Like we learned last night, Kyle's has done it for years. We talked about Annette and Seal today, but take a minute and say thank you to them, because while we all have day jobs, this is for a lot of us on this side, it's a volunteer effort, but these are their day jobs and they do a phenomenal job. So we wanted to recognize them. Okay. The first speaker that's going to join us on Finding Our Way on the Yellow Brick Road is an absolutely lovely lady named Kathy Tangent. And Kathy works for us at the stats office. Let me pull Kathy is our in operations, statistics, and finance. She is the SDMC Deputy Director. Kathy, would you join me? And can I hand off the slides to you?
I see myself disconnected.
You are
Excellent.
all, thanks very much.
Yeah. Well, I'm thrilled to be here. Thank you very much for inviting me. I usually come in a little bit later to the meeting because my meetings last till Saturday afternoon. So it was a great opportunity to come. For those of you who don't recognize the gentleman on the left, that's my esteemed colleague, Mike LeBlanc, the director of the Stat Center. And I'm Glenda the Good Witch. One of our colleagues put this together for a stat meeting. And it seemed like if there was ever a better time to recycle a slide, it would be this. So, and I'll say up front too, I'm not that comfortable with themes and memes being a statistician. So I'm doing my best here, okay? And so, yeah, in addition to helping to run the Stat Center, I've been a faculty statistician for the GU and the prevention committees for a number of years. Oops, my clock started. So I have a pretty good sense of the NCOR and the NCTN side, and I'll try to incorporate that in. And so I just kind of want to talk about how an idea becomes a trial, or at least gets to the concept stage. Okay. So for me, I'm going to be from the GU perspective, we have more ideas than we ever know what to do with. So I think the idea of a big funnel is very much how things start. We have so many ideas swirling around. It's not unusual for me to get an email on the weekend saying, I was just thinking, I mean, that happens all the time. And so there's a bunch of different concepts floating around by investigators, some of them senior, some of them junior. We tend to have monthly meetings in our committee. I think most of the committees, that's true. And then of course, at the group meeting, very typically the path for these concepts is that they're shown again and again in working group settings where they marinate, where they get input from colleagues, where they start to build a team, maybe bring in someone who's interested in the translational component of a trial. Someone else might be interested in the prose. And they start to think about these things. They start to talk about, we think about prioritizing it in the committee, but more importantly, kind of backing up is it feasible? Have we enrolled this patient population before? Are the other NCTN groups conducting studies in this group? We have to look for competition, particularly with pharma, what's going on with them as well. We look at complexity. We think about the community. Does this idea fit with something that can be done in the community? What other things I want to say? I think this is sort of where we first start talking to the companies, or we check to see if there's a CRADA so that the drugs of interest can be even obtained. I think one of the key things is the right fit for NCTN or NCOR. We can't be so fast and nimble to compete with the pharmaceutical companies. We're good at doing certain types of trials, maybe comparing drugs across different companies, or surgical approaches, or radiation, or drugs in combination. And so we think about that a lot, because the time it takes to develop a study, we don't want the idea to expire. And this is also when we start to bring in young investigators to help be mentored so they get to see how the process works. So when there's first an idea and it looks like it's going to take wings, they reach out to the stat center. We don't put too much effort in at this point, but we do get us, we have to kind of pare down and figure out some of the key questions, sort of what is the target population? Because that's going to impact eligibility, the availability of patients, so the accrual rate, who we're going to generalize the results of the trial to, and it also impacts the event rate, right? And so then we talk about the endpoint. If it's a phase 2 trial, we might want to do something short term. Maybe that's a molecular or RECIST response or PFS. If we use an endpoint like that, we want to make sure that it has some correlation with our ultimate endpoint, which is survival. So we want it to be convincing, compelling at the end of the phase 2 trial. If the phase 3 trial, it's probably going to be survival, maybe PFS, progression-free survival. But of course, that means a longer term trial. And so trying to think about how long the trial will take. So you might start off with, let's say, just making this up, advanced colorectal patients with 2 prior lines of treatment with a BRAF mutation. And then you start to look at the patient population like, yeah, maybe not. Okay, so maybe any prior amount of treatment would be okay. Maybe we'll collect BRAF status, but we're not going to make that an eligibility criteria. So you just sort of go back and forth with the team and come up with a pragmatic decision there. Another piece is the treatment effect. So I'm talking about a randomized trial. How much of a difference do you want to be able to see between 2 treatment arms? Because the bigger the difference, the smaller the sample size. So you might start off with an investigator who says, you know, in a patient population where maybe half of the patients die within 12 months, you might say, wow, if we could improve that by 3 months, that would be really clinically meaningful. But then you try to target it, and that's sort of what that bottom right corner is. 12 versus 15 months, holding everything else constant, is like 600 patients per arm. And then you're like, that's not going to work. So let's increase that treatment effect. Let's aim for kind of, maybe we always talk in baseball terms at the Stat Center, like a double or a triple. You know, with 3 months might be a single, we're going to aim for a triple. We're going to say maybe 12 to 18 months is what we're going to target. And as you can see, there's not a linear relationship there. The difference of adding 3 more months, it's a third of the sample size. So there's this back and forth. And finally, we all kind of agree on something where we've got the population, we've got the endpoint, we've got a treatment effect. And so we feel like we have enough to go forward. So this is all still kind of only at the disease committee. So then the next step is, so I always think about this, put up your dukes and fight, is we go to the task force, which is where we kind of like stake our claim. The concept is still sort of in a rough stage, but we want to sort of say, oh, SWOG is going to do this, because the next steps will take some time. Not every committee has a task force, but like in GU, we have a renal, bladder, and prostate. I know it's true for a lot of the other big committees. So even though the concept is a little rough, we go in there, leaders, let's say if it's a bladder task force, leaders in med-onc, rad-onc, surgery, they're all there in the task force, and they provide us a lot of input. And they might give suggestions about things to enhance the concept. So what we're trying to do is get to them saying, okay, you should take this now onto the steering committee. This can be a slow process, because when we get when we develop the concept and we feel like it's ready to go, we have to submit it a month before the task force meeting, which is once a month. Then when you get on their calendar, then they review it. And then it takes about a month to get those comments. Then you respond to the comments, submit it a month in advance. And you can see how 4 or 5 months can go by really quickly. But it's a little less formal. I always think of it as kind of the working group of the NCI. Oh, does somebody take out my slide? That's okay. Because I had a picture of a witch, because that's how I think some people think when they hear statistical review. I think they're like, oh, for God's sakes. But our statisticians have an incredible amount of experience, and not just in statistics. They all work across the different committees. Some of them have really good expertise at reading the study calendar or the stats section, or know how to craft the objectives just right, or great at reviewing the background to make sure it provides all the rationale and justification for a trial. And if we do that really well at this point, it means smooth sailing by the time we get to the steering committee. So we try to not reinvent the wheel, and try to think about the logistical aspects of a study. So this is a really, really important piece. Once I get the comments together and hand them to the study team, they're always like, oh, these are really helpful, thank you. So usually we feel validated for taking a few more weeks. It's a once a week meeting, so you get on the calendar real quickly. And then we go to the SWOG exec meeting. So this is sort of the leadership of group chairs, ops, stats, all the executive officers, and also importantly, the patient advocates are there on that review committee as well. This is also a weekly meeting on Monday mornings. And they talk about big picture ideas, sort of, does the trial make sense within SWOG? If there's randomization, we're talking to the patient advocates. Is this something that the patients are going to be accepting? Is it good use of resources? Does it fit into the disease committee portfolio? Like if it's a tiny single arm phase 2 and accrual is low in that committee, we might say, you might want to prioritize this a little lower and go for a bigger trial. We also talk importantly about the burden on sites. We look at the pros. We always ask how long it takes to complete those forms. We know we still ask a lot of the sites, but we try to at least have that dialogue and look for justification. Okay. We rarely reject studies, but we often have substantial review comments that say resubmit, really revamp it, but we try to help them get to yes. Okay. And then the steering committee is sort of like the exec. All the top leadership of that disease are sitting on that, including the NCI, all kinds of different experts. We go and we have an open session, talk for them with 10 minutes, 15 minutes, answer questions, and then we leave the room and it's just the worst feeling that they're going to be talking about us. It's rare that we get approved the first time. It's usually revise and resubmit, but sometimes they disapprove it. And I'm sure Crystal will talk about this idea of only 3 things on the clock, but we have to stagger our submissions of these concepts, because once the concept's approved, the clock starts running and we really everyone hits the ground. And so we have to stagger that so that we can all do it. Okay, thanks so much.
I'd like to introduce next from the Wizard of Oz. I'd like to introduce Crystal is a new protocol manager at the Center, and she has kindly agreed to join us and talk about the onset protocol.
So welcome. Thank you. Like she said, my name is Crystal Miwa. I am the protocol department manager here at the SWOG operations. I've been with SWOG now for 9 years. I've worked at a site level. I have done data entry. I've been in clinical trials now in some way, shape, or form for almost 20 years. So I am very happy to be here with you today to kind of give you an idea of what we do here in the protocols department office to get this off and going. So as Kathy kind of told you a little bit about the capsule process, we really come into this part right about when the stats are having their protocol review committee of the capsule. And so this is still right at the beginning stages, or at least I still think it's at the beginning stages. We've finally got a scientific plan. It's got some statistics information in it, and it's got some basic information that we need in order to move forward. So we put on our wizard hats and we start thinking about what this study entails, and we start diving into some of these details. We need to understand the study design. What does that mean? What is the patient flow? What happens to the patient when they come into the hospital, the clinic? How long do they have to stay there? What procedures do they have to do? When do they get to actually leave? Looking at the design of the study, how many registration steps are there? Is there 1 registration step, 2, 3? Do we need a screening step? Do we need to have a registry or a randomization step? And how does that compete to how many consents do we need for this study? What would make sense for the patients and giving them the information that they need at the time that they need it? Is this a treatment related study or is it a non-treatment related study? What kind of correlatives are we going to do? What kind of samples are we going to be collecting? What are the patient outcomes? And what kind of imaging is needed? So we really are taking all the ideas that you've been working on in the capsule development, and we're going to make it into reality. So, we start all of this going and we get out our wonderful crystal ball here and we start diving into these details. We want to understand what kind of phase it is. Who holds the IND? What does that mean for safety, the SAE reporting? Who are our collaborators? Do we have to work with different pharma companies? Do we have to work with multiple pharma companies? Where's the funding going to come from? Is it an investigational drug? How do we get that drug? Who's going to distribute it? Is it going to be CTEP or is it going to be McKesson? And so we start diving into all of these details and really kind of pulling it all together. And this is kind of really the fundamental pieces of how we create the protocol. It gives us an idea of what we need to do to get to that finish line. And so in general, it's simple, easy, right? Easy process. We take the SWOG capsule, we turn it into the NCI, LOI, or concept piece. We get approval, we do protocol development, it goes through some more reviews, and we activate, right? Pretty simple. Well, not exactly. This is how complicated it gets. And this is even a simplified version. We have to take all of those different pieces that I just mentioned and figure out what route needs to happen for each individual item. So yes, you have the protocol and that's the overarching item, but what about the imaging piece or the TM piece? What kind of samples are needed? How do you get, and so whenever I teach this with my group here, I always think of a peanut butter and jelly sandwich. And if you ever watch how to make a peanut butter and jelly sandwich, we have to think about that process, right? What kind of tubes do you need? What kind of blood do you need to collect? How much blood? Do you need to process that at the site level? How do you need to ship it to wherever it needs to go? What kind of processing needs to happen there? And then what is that end result? So you really have to think about each individual piece and put it on its own timeline and work with your different experts to really think about what needs to be included. And so there is a lot that goes into this. And like Kathy said, as soon as we get the NCI approval of the concept or LOI, this starts our OAWG timeline. Now, it may seem like a lot of time, 400 days, 450 days. We also have SWOG target days, which allows us to really kind of project management our studies, because in reality, we have targets, but it looks great on paper. It does not happen in reality. And so we need that buffer time in order to make sure that we get to these studies activated before the drop dead date. And so what we do is we take a lot of that development timeline and we break it up into different parts. And you can see here, I've got it labeled part 1, 2, 3, 4, and 5. And we separate out that journey or the number of days for each individual part. Okay, these are our target timelines. In reality, it doesn't happen, but this gives us an idea. It's able to help us predict how long it's going to take us to get to the next milestone in this study. And so within each of these, we really kind of dive out and have an idea of what we do for each of these pieces. So for part 1 and 2, and I kind of merged them together to keep it simple, we've got committee approval. We need to have our different committees, our committee chairs, the experts in our committees approve any type of design that is going to go into it. We take that information, we figure out what our study assumptions are. Is this study actually feasible? What are the pitfalls that need to be done? So we do our risk assessment. We need to get that firm understanding of that study plan. And so once we get to protocol development, we're actually creating the protocol. We are getting down to the nitty gritty. And I have to tell you, sometimes a single word will affect a lot of your protocol. And we get questions from sites because we messed up on a single word. So we really have to read it, understand it, and put ourselves in your shoes and the patient's shoes of what makes sense for our protocols. We have multiple reviews for these documents. Our first draft has lots of holes in it because we don't know the answer. We're looking for those answers from our statisticians, our study chairs, our farmer groups to kind of grab all of this in. And so then we get to our rapid. If you've ever participated on a rapid, it's a 4-hour call. It's very long and drooling, and sometimes we have to have 2 of them. And we really need to go through the protocol page by page to understand if there are any red flags. Does this logistically work? Is it still scientific? And so we have to go through each of these items. Once we go through the protocol, then we go through the consent, and it takes some time. And then of course we've got another stats PRC for that review as well. And they, again, do a lot of the feasibility. It's kind of their kickoff on how to do the form designs and everything else. And then we finally are able to submit it to CTEP or DCP for their review. And of course, they're going to have comments. So this all takes time. A CTEP review can take, usually it's about 2 to 4 weeks. We have to respond to those comments. We're hoping to respond to those comments in about 2 weeks. And so the CTEP CIRB process takes on average about 3 months. Okay, that's on average. And then we also work on, during this entire time, we're also doing the electronic data capturing form creation as well. And so that process is also taking place. And then hopefully by the time we get to activation, we've got drug where it needs to be. It either has to be at the PMB or it has to be at McKesson. Hopefully that's done. Any contracts or budget executions that we need to do. Are our labs in place? And so really thinking about each of the individual pieces in order to make sure that we can activate the study. And if somebody's going to put a patient on study that day, everything is in place. Okay. And this really takes a lot of collaboration from everybody at SWOG. We get information from you, we get information from stats and the study chairs and everything else. So it's a big group that we work with to make this dream come true. So we can dive deep into our crystal ball, or we can get into our basket of experts in working on this project. So thank you.
So we're delighted to have a very special presenter join us this year. Dr. David Zen is an assistant professor and gastrointestinal medical oncologist at the University of Washington Fred Hutch Cancer Consortium in Seattle, Washington. His specific clinical and research focus is in neuroendocrine neoplasms and gastroesophageal cancers. His research is focused on identifying new therapies through clinical trials, incorporating translational research with the goal of identifying biomarkers that better predict response to therapy. He is currently the national PI for SWOG 2012 in neuroendocrine carcinoma, which is a very practical and outstanding learning experience of what this is actually like. We really appreciate he's juggling between different meetings and he's taken the time to do this with us today. So please give a very warm welcome to Dr. David Zen.
Thank you so much for doing. Well, good morning, everyone. And I want to thank you to the ORP committee, as well as Maggie So from my institution for inviting me to speak to you today. This sounds like a fantastic talk, and I would say things that Crystal and Kathy had alluded to his talks. I mean, I was just like going through all this, like I went through that, I know this. So, and hopefully this will give you a real life example of what I had to go through for this study. So I'm going to be talking about my study, SWOG 2012. And so the outline for my talk is really focusing on the personal experiences and just kind of what we went through. And it will highlight a lot of the steps that Kathy and Crystal spoke about as well. And I actually learned a lot from this process and also helped a lot with my professional development, which we'll touch base on as well. Now, I think Crystal mentioned how long it takes through all these steps. She was not kidding how long it takes. Okay. And so this started in 2018. So this is over many years ago. And I don't want to make this talk as like a really scientific talk, but in order for us to actually get through this study, I do need to give you some basic knowledge about neuroendocrine cancers. And that will be very relevant in terms of some of the obstacles that I face when I presented the study within SWOG, but also at the NCI. So what are neuroendocrine neoplasms? So neuroendocrine neoplasms, these are cancers of neuroendocrine cells. So neuroendocrine cells are actually all over your body. And what I tell patients is that these are the hormone producing cells. So they make chemicals, and these are your chemical messengers of your body. That's how each of your organs speak to each other. So you secrete thousands and hundreds of chemicals every day. Just like any cell in the body, though, they can become a cancer. And we've come a long way in terms of how we classify neuroendocrine neoplasms, and in a very broad way, at least in GI. It's based on the WHO classification. So we think about things as one, the first step is the differentiation, meaning when we look at the cells under the microscope, they all have to come from parents. So do the cells look like their parents, or have the cells changed their behavior so much that they become more aggressive? So something that still resembles like their parents is a well-differentiated neuroendocrine tumor or NET. Something that's really aggressive, does not have the same facial features anymore, is a poorly differentiated neuroendocrine carcinoma. And that's the purpose, and that's the focus that will be for SWOG 2012. And then we also want to kind of get a sense of how quickly the cells are growing. So the other thing that we get from pathology is something called a proliferation index, meaning how quickly are the cells dividing. And so we get that by a special stain called Ki67. In neuroendocrine tumors, we have different levels depending on how quickly they're growing. In neuroendocrine carcinomas, they're all fast growing by definition. But there's two types of cells. There's small cell and large cell. And that just refers to when we look under the microscope, they look like small cells, they look like large cells. That's really the two types that we have. And why this all matters from a practical patient perspective, and especially from a design standpoint, is that neuroendocrine tumors and neuroendocrine carcinomas are very different. So what I have here is that on the chart on the left, basically the top bar is the group of patients with slow growing, well-differentiated grade 1 or 2 neuroendocrine tumors. This is a survival curve. And so here is the bottom of months. So what you can see here is neuroendocrine tumor patients can actually live many years and sometimes even decades, even with stage 4 cancer, okay? Versus a neuroendocrine carcinoma in this red line, these are patients who live less than 1 year, okay? So why it's very important that even though the terminology is very confusing, it's very important for us when we're actually counseling a patient and talking about what are the treatment options. And one of the things that we've learned over the years is that I didn't really highlight it in the last table, but there's this well-differentiated grade 3 group. So that's a group of patients who have fast-growing cells, but the shape of the cells still look pretty well behaving. And in the past, we actually used to lump them with neuroendocrine carcinomas with the very bad actors. And we actually learned we were wrong in doing that, because what we found is that this group of patients in blue, they're actually somewhere in the middle. They're not as good as the ones who are really slow growing, but they're not as bad as the ones that are fast growing. And what we've also learned is in the past, we used to treat these patients like a neuroendocrine carcinoma using very aggressive chemotherapy. And what we're learning now is that chemotherapy actually doesn't really benefit them. We're actually just giving them more side effects. So from a trial standpoint, when we actually designed this trial, we actually learned about this and said, when we think about that, how are we going to include patients or exclude these well-differentiated grade 3 patients and make sure this is a very relevant for a very aggressive type of cancer? So when we talk about neuroendocrine carcinomas, this is the poster child of what a SWOG and NCI study should be. This is a rare cancer, okay? So when we focus on neuroendocrine carcinomas, what I have here, this was done by Dr. Desaria, MD Anderson, a very nice review of neuroendocrine carcinomas. So on the top panel A here, this is a pie chart where vast majority of neuroendocrine carcinomas start in the lung. That's what we call small cell lung cancer, okay? But if you go from this green pie, which is extrapulmonary, which is our term saying everything that occurs outside of the lung, now we're talking about a small piece of the pie. And if we break that pie down further, actually a good chunk of it, about a third, is actually GI. So that's actually what we see in clinical practice. But other sites do, but these cancers do occur in other sites. And on the pie chart here in B, you can see GU tract, gynecological tract, so really all over the body, because these are cells that are over the body. And then just to give you an idea of the prevalence of the different types, I mentioned neuroendocrine carcinomas, there's small cell and there's large cell. In the orange bars, these are small cells. So this is the prevalence of, if you look at GI, if you look at lung, if you look at other sites, versus blue, which is large cell. And you can see in most parts of the body, small cell is the predominant histology, with the exception in GI. In GI, we actually see a little bit more large cells than small cells. And this will come into an issue in terms of when I design my study. So how do we treat neuroendocrine carcinomas outside of study? So this is a very aggressive cancer for which we really did not have any prospective studies because it's so rare. So it was really hard to design a study when out in community practice, you may see only 1 or 2 of these patients per year. Okay, that's how rare it is. Okay, and so really a lot of our data was based on retrospective studies, and we actually copied our lung cancer groups because one of the shapes of the cell is small cell, and say, well, it looks like small cell lung cancer, so let's treat it like small cell lung cancer. And how we treat small cell lung cancer is with chemotherapy, which usually has a platinum agent, which is usually cisplatin or carboplatin, and another agent called etoposide. And for many years, everything was retrospective studies until 2022 at ASCO GI, we finally had the first prospective study done in Asia, where we actually now have real-time data. But no matter what trial you look at, the survivals are poor. So in this column here, you can see it's about 12 months. So that's how poor it is, and that's how we've been treating patients for many decades. So obviously, there's a lot of room for improvement for this group of patients. Now, in lung cancer, we have new agents. We have immunotherapy. Everything's about immunotherapy and checkpoint inhibitors. So what about immunotherapy? Well, if we extrapolate our management from small cell lung cancer, well, what's the data in small cell lung cancer? Well, in small cell lung cancer, if you use checkpoint inhibitors by themselves, response rates, the chances of shrinkage is pretty low, so on the order of usually less than 10%. Progression-free survival is just about 1 to 2 months, and survivals is pretty poor. It's just about an order of 7 or 8 months. And so obviously doing single agent immunotherapy is not going to move the bar. And likewise, there's been some small studies looking at extrapulmonary. So in other words, if we see neuroendocrine carcinomas outside of the lung, what's the data? It's pretty much the same in small cell lung cancer. So as I was kind of thinking about concepts about how we move the field forward, and we know that we were using immunotherapy in small cell lung cancer, one of the things is based on the prior data is that just using immunotherapy by itself is not going to work. So we need to think about other approaches. And this is where one of the SWOG studies actually provided a basis. And this was an ongoing study. This is, and many of you may know, the DART study, SWOG S1609. And this was a very novel study in that this was a study dedicated for rare cancers. And the definition of rare cancers was it had to meet a certain incidence. And this was not just for neuroendocrine cancers. This were for many different types of rare cancers. But they did have a group of patients that actually had neuroendocrine cancers. And one of the early data that they actually demonstrated using combination checkpoint inhibitors. So not just one drug, but 2 drugs, nivolumab and ipilimumab. So some of the early data, what they demonstrated, so I showed you that WHO classification. So they included a lot of different types of patients, neuroendocrine tumors, the slower growing ones, neuroendocrine carcinomas, the faster growing ones. One message that we took home from that study, it was only the faster growing ones who seemed to get some benefit to immunotherapy, not the slower growing ones. So obviously that was the first basis saying when we're designing the study, we really need to focus on these really faster growing cancers. The second is we actually started seeing shrinkage, unlike the prior studies where 1 drug, now with 2 drugs, we actually do see some benefit. So the take home we got from this study when I was kind of proposing my study is that, okay, it looks like we have to think about combination treatments. We can't think about single drug treatments. And so along that lines, that study was basically after patients had progressed on prior treatments. And obviously we know in clinical practice, the earlier that we move a treatment early on, the better that we can potentially improve outcomes for patients. And in small cell lung cancer, they actually had a study that demonstrated when they combined standard chemotherapy, the platinum and etoposide, with an immunotherapy agent, atezolizumab, they could actually benefit survival. And this came from a study called Empower 133. And the take home from this study is that when what you have here is a survival curve on the left. The top bar is patients who got carboplatin, etoposide, and atezolizumab in newly diagnosed advanced small cell lung cancer versus if you just got chemotherapy alone, okay? On the table down here gives you the median. So the median overall survival, there was about a 2-month improvement if you added atezolizumab, which is not really significant. That seems, not great. But if you actually look at the survival curves, it's the chance of having a greater chance of living a longer period of time that actually was pretty meaningful. And so because of that, in small cell lung cancer, atezolizumab was actually approved in combination with chemotherapy in 2019. So that set the stage to say, what about neuroendocrine carcinomas? Could this do this? And actually people in clinical practice was doing that off-label, but without any sort of data about whether that would be beneficial. So that's where along with my mentor at my institution, Dr. Kiorian, who many of you know is a leader within the GI field, we proposed this study where basically very small study, and we recognized this was a very rare cancer. So we wanted to get information, but we also want to do it quickly. So we thought, okay, well, let's do a phase 2 study. Let's just get some information. And we would be very broad. So we would allow patients with neuroendocrine carcinoma, small cell, large cell, doesn't really matter. Any site that's outside of, or actually this was really focused more just for GI because we were both GI oncologists, and just pretty much recapitulated the study that small cell lung cancer did, which is 4 rounds of chemotherapy with immunotherapy, and then 1 year of immunotherapy if your cancer didn't progress after that, versus the control would be just several rounds of chemotherapy by itself. And we had a primary endpoint of PFS, because just like what was discussed earlier, overall survival, you would probably need more patients. And so we presented this within the SWOG GI committee. So just like what Krista was saying, of discussing within your disease groups, we had a lot of discussions about this, right? And everybody was pretty favorable. They said, yep, this is a rare disease. We have not made any headway. We really need to do something. But accrual was the concern. It was always about the feasibility, and we weren't sure how we were going to do this well. And the other thing is ECOG actually had a competing study. So they were actually doing a neuroendocrine study, but they were including well-differentiated grade 3s. So that group of patients that I said shouldn't be getting this type of chemotherapy, and large cells, but they didn't include small cells, right? And that, I won't go into details about that, but that created some nuances that the NCI was going to take into consideration. And the question was, well, what's the endpoint we should be doing? You know, progression-free survival is good, but really it's about how long do we keep patients alive, right? So should it be overall survival? So SWOG GI said, okay, well, let's go ahead and move this forward. Let's have the neuroendocrine task force review it. And I think at the neuroendocrine task force, I gave you this weird face because it was good. Everybody was like, yeah, we like your study. We agree it should be done, but really, how do we get these patients? And then it was like, how do we make this a meaningful study? Because if it's going to be a rare patient population, and you're going to take a while to get a study done, we won't be able to do the study probably again. So you might want to get your answer now. So if you want to be definitive and say, this is a treatment we should be doing forward, you need to have a phase 3 trial. But at the same time, we don't want to commit to a phase 3 trial if the whole trial is going to be negative. So they came up with like, could we do a phase 2/3? So in other words, you start to get some data initially. If it doesn't pan out, you don't do the phase 3. But if there is some signal of activity, then you can move to the full-fledged trial. So they recommended doing phase 2/3 trial. And again, that would be a bigger study, but I didn't have a problem with that. And I agree completely, we need to avoid any sort of barriers. So no central pathology read, allowing 1 prior cycle of therapy prior to treatment, and that all made sense. But then what didn't really make a lot of sense to me from the NCI was, you told me accrual is a concern, but now you really just want to restrict to small cell only. And there were 2 reasons. One, they said, well, if you have small cell and large cell, maybe there's some differences. It'll be too difficult to kind of interpret the study. The other was the ECOG study, really, was that we didn't want to have competition with the ECOG study. So essentially the decision was made that, okay, well, why don't you revise the study, but you would have to restrict it to small cell. And at the same token too, because now they also said, well, we don't want to focus on GI. We really need to do all the body systems. Well, you're not just GI, so please go to the early therapeutics committee. So now I'm going to a committee that I've never worked with, right? And had to present to a new group. So a lot of processes here that were involved. And so this was the revised schema where now we're doing a study where it's all things outside of the lung, not just GI, but very restrictive to only small cell only. And now we have a primary endpoint of overall survival as well, and we're allowing 1 cycle prior treatment. And initially, I showed you my initial schema had a accrual of about 30 to 40 patients. Now that jumped to 134 patients, right? And we're talking about a rare cancer. So we get there, and finally, we go to the executive review committee. They approve it. I was very happy. And finally, we get a name, SWOG 2012. But little did I know that for 1 year, would this be me just bumping my head against the wall? Because eventually, when we get to the final review with the NCI GI Steering Committee and CTEP, again, everybody said, yep, important study, but accrual was kind of the concern. But then they also brought up this point, well, like the data that I showed you, single agent immunotherapy doesn't really provide a lot of activity. There's been no study to demonstrate what's that impact of giving that extra single agent immunotherapy after you finish the several rounds of chemotherapy. And so, because on all the small cell lung cancer trials, they just did it automatically. And they were like, we want data on that. And so basically the GI steering committee said, we're going to reject your study because we now want a 3-arm study, not a 2-arm study, because you need to answer the question of what is that benefit of the single agent immunotherapy. So went to Roche Genentech, who obviously was supporting to provide atezolizumab, and they basically said flat out, no, we're just not going to approve the study. And from a company standpoint, if that arm, if you don't give the 1 year of immunotherapy afterwards, it's like less financial impact for them because they don't have to give any more drug, right? And so they saw that as a downside. CTEP said, nope, we're going from a scientific basis. We want a 3-arm study. So basically it was back and forth, trying to appease both parties. And I think where we kind of all came together was that, look, let's think about the patients. This is a rare patient population. We are going to get nowhere if we don't do something different. So even if it's a bigger study, if this is the way that we have to do it, let's just get it done. And I think that's really was the take home message. And so finally, after a lot of negotiations, we all came to agreement that we would approve a 3-arm study. And this is now the final version of the study. So essentially you can see how it's evolved over several years. Instead of not just 2 arms, we have 3 arms. And the 3rd arm is this red arm where we do chemotherapy and immunotherapy. And then if you don't progress after 4 rounds, you just go for observation, okay? And that's really trying to answer the question of, in this other experimental arm, what's that benefit of giving the 1 year of immunotherapy by itself, okay? And again, same design, all extrapulmonary neuroendocrine carcinomas outside of the lung. It was still restricted to small cell though at that time, and the primary endpoint was overall survival. And this was activated December of 2021. I said I started in 2018 when I presented to the GI committee. So we get it open. I'm so happy, but I knew the small cell restriction was going to be a tough thing. And it was a tough thing because we only enrolled 3 patients in 1 year. And we were actually on the docket on the NCI of a red flag for basically a low accrual. But the things happened kind of for a reason. And during that time, the ECOG study had already closed. They had presented their data. And so that provided an opportunity now that there was not a competing study within the NCI, within the NCTN cooperative group. So basically modified the study, amended the protocol. We got that amendment just actually approved January of this year. And now from 3 at the end of December 2022, we are now at 11 as of today. So basically now we're finally meeting the original estimate of about 3 patients per month. So you can kind of see that there's many things where you kind of start out, but then after a lot of input where everybody has, a good heart for it, sometimes it just really creates a lot of obstacles along the way. Now, even though it was a lot of work, I think at least for me, I did have a lot of professional development for that. So because of a lot of my involvement, and this was even before the study finalized, I was elected to be a SWOG champion. I'm now on the NCI Neuroendocrine Task Force as an early career member. I'm in the NCCN guidelines for the Neuroendocrine Adrenal Guidelines panel. And I've also built a neuroendocrine program at our institution. And so just because a lot of that, it's just the professional development, right? Because even if you do a lot of work and even if things don't really happen, you actually get recognized by people. They're like, okay, this guy is actually putting in the effort. He actually wants to do this, even though he might know that the reward might not happen at all, right? And so that's really what really helped me. And I think that's really what I've taken a lot from being part of the NC10 cooperative groups. And one question I get from people is, would I do this over again? And interestingly, yes. I will say that, every time I go to the barbers, I notice quite a few more gray hairs, and I'm really not that old yet. So, but I do blame a little bit on SWOG as 2012. So, but I will say that, I've grown a lot, both personally and just professionally through all of this, and just a lot of networking opportunities, both not just with GI, but just also in other disease fields as well. And so part of this, at the end of the day, it's really just trying to improve the lives of our patients. And so, yeah, it's just been a wonderful experience despite all the obstacles to this.
Thank you, Dr. Zen, for taking us on your journey. We're going to break now for about 10 minutes, and we will resume down the Yellow Brick Road at around 9:05. All right, thank you. We're going to get going again.
I'm sure you know who I am. But in case you didn't get the memo, my name is Glinda. I am the Good Witch of the North. As you can see, my picture's up there, and I'm with Dorothy. And all right, so it looks different. That's what research does to you. makes us a little older, a little wiser, a little blinder, a little whatever. But anyway, I'm going to help you on the next part of your journey if I can walk around in this dress still. Okay, so we have a rule. No posting Glinda pictures on social media. Because Glinda has to go back to her day job, just so you know. Okay, so on our line, we have a wizard from the NCI. I want to tell you that we are extremely blessed with both Wizard Andrea Denikoff and Wizard Grace Mishkin. And today, Wizard Grace has been able to share some time with us. And she is going to take us on the next step of the journey on the Yellow Brick Road. Grace, are you there?
I am. Can you hear me, Glinda?
We can, and I'm so
Wonderful.
grateful you're joining us today. I'm so excited. Anyway, would you tell us a little bit about what it's like when the protocol gets to the NCI?
Absolutely. Next slide, please. And I'm seeing a network issue and not seeing the slide advances.
Well, you know, that's the moment
Yep.
that Glinda solves all problems.
Glinda solves all
Just
problems.
like the
So
researchers do, right? Just like
Exactly.
you do. Glinda
I can-
will assist.
And so I can go ahead and start talking. Wonderful. And you can go on to the next slide, key NCI rules. And so I am speaking on behalf of myself and Andrea Denikoff, who I think everyone knows is more of a wizard than I am. And we are grateful for the chance to talk with you all because we're very sorry we couldn't come to the meeting in person, but we are delighted that we get to share a bit of the NCI side. I know some of this will be overlapping information that you're hearing from other perspectives, so I won't dive too deeply, but just to give you an idea of what happens at the NCI side. So the next slide, please. The first thing to know is that, yes, at the NCI, in addition to all of the acronyms that your institutions and SWOG uses, we have plenty of our own acronyms. So our clinical trials that are on the treatment, imaging, and biomarker-driven side are funded through NCI's Division of Cancer Treatment and Diagnosis. And specifically, Andrea and I and the NCTN are in the Cancer Therapy Evaluation Program. On the other side of the literal building at NCI, on the other side of our big building, the Division of Cancer Prevention supports quality of life endpoints in NCTN trials and also supports cancer prevention, screening, symptom management, cancer control, and CCDR trials through the NCORE. And so we are in two distinct divisions, which can be the cause of some confusion. It's part of why sometimes something can happen in an NCTN trial and not in an NCORE trial. And so just to let you know that those are two very distinct entities at NCI, although we work very collaboratively together. And we do apologize when there ends up being confusion because of our two sides of the building. The one other set of place within NCI I wanted to let you know of was that the Coordinating Center for Clinical Trials oversees the steering committees that review concepts. Next slide. So NCI has some involvement even before there's a formal concept or LOI being developed. There's parts of CTEP that work with industry partners and maintain a portfolio of the investigational agents that can be used in trials if they're being conducted under CTEP's IND. And the CCCT steering committees and task forces are discussing trial ideas and disease portfolios before early in the development of a potential trial idea. Next slide. But then the real excitement obviously starts when your group has developed a letter of intent or a concept, which is just a short overview of the trial idea with a bit of key information. The LOI or concept comes into NCI, and if it's an LOI for an early phase trial with fewer than 90 to 100 patients or participants, then it's reviewed internally by NCI. If it is a concept for a later phase or larger trial, the decision is made by one of those steering committees. But either way, that document is evaluated by reviewers from across NCI. There's disease experts, experts in that agent portfolio, biostatisticians, and others, depending on the particular issues being raised in that trial idea. Next slide. And that initial review of the LOI or concept is the start of the deadline. I know you saw some information about the OEWG timelines, operational efficiency working group timelines. And so from NCI's side, the review starts when that LOI or concept is reviewed, and then you have 400, 450, or 540 days to get the trial activated from the time of that initial review. And so once that LOI or concept is approved, unfortunately, some are disapproved, but we're talking here about the ones that are approved, and the group starts working on developing the protocol. It must be based on that approved trial design that was in the LOI or concept. And then all of the work is towards getting that trial to activation. Next slide, please. NCI isn't as involved when the group has that approved LOI or concept and then is working on the protocol, but then the group submits that protocol and the consent documents, any related documents, and all of those documents are reviewed as a package by a really large group of NCI experts. And so I wanted to highlight here that there are a bunch of different parts of NCI and areas of expertise at NCI that are involved at the protocol, starting at that stage. And there actually is always a full discussion. We have a weekly review meeting here at NCI. So it's actually every Thursday morning, we have a meeting that lasts typically 3 hours, sometimes up to 5 hours, that involves all of these reviewers where we review the latest LOIs, concepts, and protocols. And so those reviewers that are reviewing the protocols and discussing, presenting the issues, and ultimately informing the decision that's made include those disease experts, agent experts, radiation oncologists, biostatisticians. We have regulatory specialists, pharmacists, imaging experts, translational scientists, and quality of life reviewers. And I will say the one piece NCI is aware that we don't have as much input or feedback from is your site staff. We really rely on you working with your group because we don't, other than Andrea and myself, and I unfortunately do not have site expertise, we don't have a formal review process of reviewing it from the site perspective. And so I think, as we've heard, more and more of that is being done at the group side, and I think that will be increasingly valuable. Next slide. So once that initial protocol is reviewed, almost every protocol, at least a few changes are going to be required. CTEP, Cancer Therapy Evaluation Program, is communicating with the lead group and the study team to provide the revisions that are needed until a protocol and consent is finalized. And then Andrea and I work on the funding sheets. They were also submitted at this time of protocol submission. And based on the final approved protocol, we approve the funding sheets with the final funding amounts, both federal and non-federal funding, depending on what the group has obtained in terms of non-federal funding. If the study is under CTEP IND, then we're obviously playing a more intense role throughout this process. CTEP would be the study sponsor, coordinating with the FDA for all of the regulatory submissions and coordinating with the company for any non-commercial drug distribution that's happening under CTEP IND. Finally, when the protocol and consent have been approved by all of the NCI reviewers, it's then sent to the CIRB for review. Again, typically there's at least one round of revisions required. And then throughout this entire process, CTEP, NCI's federal staff are also working closely with the contractors that are working with the lead group to set up the study in all of the CTEP systems. So we do have a lot of centralized support that's provided through contractors like the CTSU, like Open, doing things like developing the support documents. So thanks to the efforts from many at SWOG and others across the groups, that included developing the national coverage analysis documents a few years back. And then we're very excited that this spring, we are now also developing EMR templates. That's being done by that CTSU team. And so those CTEP contractors are working with the lead group and NCI to make sure that they have the appropriate protocol document to be developing all of those documents. So there's a lot of back and forth happening. Next slide. And so I'll end with just 2 slides talking about how NCI's role doesn't end after activation, just like, the yellow brick road keeps going in my imagination. So After protocol activation, we continue to review all of the proposed amendments, and all of the reviewers from that previous slide are brought in if any amendment affects any section that they are primarily responsible for. Sometimes there might be amendments that we request from the lead group. There's participation by NCI experts on the group's data safety monitoring boards for the ongoing monitoring of the trial, updating things throughout this contract systems and funding sheets as needed when there are changes to the protocol. We do track accrual using specific slow accrual tracking rules for the first 2 years to make sure that the trial is feasible and accruing appropriately. And that's based on the original plans that were submitted by the investigator, and generally ensuring compliance with regulations and NIH grant requirements throughout the activities of the protocol. Next slide. And this is my final slide, is just that there are more intense activities involved for CTEP IND trials. So there's some level of monitoring for the adverse events, continuing to distribute the drug, the IND agent, coordinating with the company, submitting reports, and generally completing all activities that are required of an IND sponsor. One thing I wanted to mention here, because I think it's a question that comes up some, like I said on the last slide, sometimes we have to request amendments. And this actually goes back to, issues that were raised once upon a time by, to make sure that we were accurately and adequately tracking if there were any changes to the adverse event profile of the agents that are under our site CTEP IND. So we're actually required to see if there have been any changes to the risk profile of the drugs on at least an annual basis. And then if there have been, that's why we have to request amendments to the trials to make sure that that updated risk profile is included. And so I know that is one of those areas that often can be challenging from the sites to have to implement those amendments. We know that it's challenging, especially when it seems like the changes are relatively minor. And so, we're kind of working within the same regulatory framework sometimes that you are, trying to do the best that we can to get these protocols through and keep them as productive and feasible as possible. And so we do apologize when there are issues at our end that end up causing more work at your end. We're much more excited when we can find ways that can hopefully make your lives easier and make the protocols more successful throughout the system. So I am happy to hand it back to Glinda. I know you're in wonderful hands, and thank you all for everything that all of you do to make these protocols so successful, because everything that happens throughout the other aspects of the system wouldn't matter if it wasn't actually getting to the patients. And we know that that's only possible because of what you're doing. Thank you.
Is this wand working? Oh. Wizard Michigan, that was spectacular. I cannot thank you enough for zooming in from the stratosphere. Thank you again. Now, there are lots of supportive friends along the way. You all know that. You're all very supportive of each other. But there's a special group in SWOG called the Disease Liaison Committee, and I just want you to be aware of them. There are 2 new chairs, Sandy and Aaron, and you saw their names up on a beginning slide, but I'd like you to get to know them because they represent all of the disease liaisons. And as you can see from the slide, they offer feedback, they help with development, they involve site perspective and operations, which we all know is incredibly important to make it actually work, including the wand. And they keep the lines of communication going. So I think it's really important that for those of you who are interested in this particular area, wait, there, I see there's liaison over there. I'm sure there's one over there. And I know there's one right there. We need some people. And I'd like you to take note of this. There will be people around at Open Forum who can take your level of interest, particularly Auntie M, particularly my co-partner in crime. And so please let us know if you're interested. Now, we're going to talk a little bit about feasibility because you know what? You can't bring every protocol to your site. You know that. Okay, there are protocols that you don't see the disease group, you don't see the people. So I would like to call our next set of speakers up, Ashley Tyden, the Deputy Director of UC Davis Cancer Network, Anthony Hicks, the Operations Supervisor at Cancer Research Consortium of West Michigan, and Ms. Rachel Kitchen, Senior Regulatory Affairs Coordinator at UC Davis Cancer Care Network. And they are going to talk to us about how we look at assessing interest and relevance. Thank you so much, Ashley.
Thank you so much, Linda. So wonderful to be here with you. We're so grateful to be here and talk about the feasibility processes that work for us at our sites. So Rachel and I work at UC Davis. We're a lab site and a main member of both SWOG and NRG. We specifically support our cancer care network, which are community-based cancer centers that are our affiliate and integrated component sites. And Anthony works at an NCOR with 10 sites, and he manages the regulatory affairs for his entire consortium. So identifying those trials that best fits your patient population and are actually doable is so important to making sure you have a balanced portfolio and your trials run smoothly and ultimately are successful. So I'm going to ask our panel a series of questions we developed that highlights our best practices, our brain trust, and some of actually the key differences in how we manage this process. All right. Images that Glinda picked for us. So first of all, how do you monitor the availability of new trials and assess interest? Anthony.
Yeah.
Push the button.
Oh, no. Oh, okay. There we go. All right. So how do we assess interest in new trials? From an NCORS perspective, we monitor all activated NCTN clinical trials, whether it's through the activation emails or CTSU. How do we stay organized, you may ask? I have a spreadsheet for that. And then assessing interest, I send out interest emails to all of our providers and usually the site lead, just asking for interest. And usually they get back to me, I update my spreadsheet. And then from there, all the endorsed trials, we then go on to the next step of feasibility where we look at every aspect. So we look at the clinical aspect, we look at regulatory, we look at pharmacy, financials, specimen management, you name it, we look at it. So.
All right. So I manage it in largely the same way, reviewing those activation emails. I also search the CTSU website at least once a month for all the new trial activations to see which trials might potentially match our patient population. I also keep my ear on the ground on where we might have gaps in our portfolio, where we don't have a trial to offer a specific patient population, and try to search those trials out. So trials that match our general patient population get sent to this monthly committee meeting that's attended by all of our community oncologists and all of our research staff members. They debate the scientific merit of the trial, what our accrual potential is, big ticket logistical issues. We haven't done that in-depth feasibility review yet, so we're just kind of talking about big problems we might foresee in the protocol. And then they ultimately decide which ones are going to move forward to our feasibility process. About 20% of trials don't make it through that first round, and we decline participating in them from the very beginning. So once they're approved by DTC, Rachel does a little more work on monitoring the accrual potential. We're going to talk about that a little bit later. Okay, how do you assess your site's potential to accrue to new trials?
So from an NCOR perspective, potential accrue is very important. As you know, NCOR has their goals of accruing those patients, but part of our mission is to provide access to the latest cancer research for anyone in our community. So, you know, we may open trials that we may only accrue one patient, or we don't accrue at all. But as you heard from Toby's story last night, I think it's very important to have those studies open. But I also wanted to note too, when I send out those interest emails to physicians, we normally get an accrual estimation, so we take that into account. And then I did want to note also that we have a rare list on our website where we look at the rare studies where we may not accrue. We put a list on our website where we call it just-in-time studies. So our physicians know about this, so they know that we can get the studies open for anyone that walks in the door within a couple of days. So.
And this is one of actually the key differences that we noted when we were preparing for this panel. The differences in the way Anthony managed this is for the NCORE, and the way we manage this for our NCI designated comprehensive cancer center. So we're like heavily scrutinized on the trials that we open and whether or not we're demonstrating progress in accrual. If we're not demonstrating progress, we need to come up with a plan to improve it. If we're still not demonstrating progress, they really expect us to close those trials down. So this, again, is like one of the most important criteria that we think about. We don't want to put all that effort into opening a trial and then have to close it a year and a half down the road. So me and our medical director analyze this cancer registry data that I request from our sites on an annual basis. This is an example of a standard site table that you can receive from your cancer registrars. This data is not perfect. It's at least a year in the rears. And it doesn't have a ton of specificity on the type of patient populations that come in, but at least you can tell what newly diagnosed patients are walking in your door every year, including their stage of disease. And then you can compare that to eligibility criteria and further deduce accrual potential based on like subtypes of cancer patient populations, like HER2 subtype in breast cancer patients, for example. So this, like I said, not perfect, but such a great way to make some data-driven and informed decisions. So I'm going to talk to Rachel now about some ways that she assesses accrual potential after that committee approves the study.
Yes. So our disease team committee approves the study, and then the first responsibility is typically mine to reach out to the site. Usually we have a primary site contact. We kind of have a mix. Some sites choose to have one primary contact that I would reach out to for all of the studies. And then other sites have multiple champions that we can reach out to, and they can give us a more comprehensive view of what they think they could actually accrue. So my job is to reach out and make sure to determine how accurate the accruals are. We ask the sites to base that off of the cancer registry, just like Ashley mentioned. That's a really great resource. Also, some EMRs like Epic have patient lists that you can pull up and get a kind of basic understanding of disease types that you're seeing commonly at that site. And then also just group discussion. So we encourage sites that have the time, if they have time before we have to start the study, to get together and talk amongst the physicians. You know, we all know that some doctors see primarily breast and their counterpart might see primarily prostate. So having that universal agreement across the site that they are interested in the study and that they really do think this is a realistic expectation makes it much more helpful for us. And then like Ashley was saying, we do answer to the protocol monitoring committee. And so we need to make sure that our accrual estimates are actually what we are seeing being accrued. And then my job is to validate that information. So I assess the estimates that they provide. Again, I can check the cancer registry, the accrual history at the site, and looking at previous studies that they've opened to make sure that that number is actually what a realistic number. That's great.
All right, now we're going to move on to kind of meet our presentation here, and what are the common barriers or feasibilities, and then how do we address them, and most importantly.
So from our perspective as an NCORE, I listed here some issues, some feasibility issues that we've noted. The first one I have is no physician interest. So whether that's a competing trial or they don't see that population in the community. And then I have intense credentialing. An example I have for that is the BRCA-P Alliance study, where there's a 3.5-hour training. There's a DTL, there's a lot of other regulatory documents to submit in the background. So just a lot of intense credentialing there, or study complexities. And I want to highlight the BN-011 study there. There's multiple vendors, whether it's pharmacy or specimen management. And along with that, then you have the very strict timelines and multiple steps within the protocol that really complicate things for our staff and the patients. The next thing I have for common feasibility issues is financial or coverage analysis issues. And I would say this is our biggest feasibility issue as an NCORE, because and it's mainly those scans. Scans are expensive. So, and we don't want the patient to be held accountable to that either. So a lot of times we don't open those trials because we can't cover the cost either for them. And then the last thing I have here is protocol issues identified. So whether the protocol and informed consent aren't consistent. And the example I have for that is an Alliance study A08-2002, where the quality of life and blood specimens were inconsistent between the two documents. So as you, so how do we address them? We actually emailed the study. And the best success story I have is the last study I mentioned, the A08 2002. We notified that study and they did create an amendment and fix it. And we are in the process of opening that study currently.
With our list of common barriers and feasibility issues, first, logistical concerns. Sometimes in community hospitals, you just don't have the subspecialties available. You may not have the radiation techniques that would be required. Even local surgeons, we had at one location where there wasn't a urologist in the area, so that was a challenge. So it's really important to look at those kind of logistics to see if that's something that would affect your site. Also, something to consider is the financial burden, not just for patients, but just starting with the their driving, transportation, hotel costs, all of that, daycare, those are all things that patients have to consider that can really affect whether or not they can participate in a study. And we oftentimes can't support that. So we have to look at that kind of burden to see how we can help the patients. We also will look at the coverage analysis and compare that to standard of care for the site to see if that's going to be an issue that we would be concerned with. So that's something that really should be kind of a first check, is to make sure that financially can your site handle it. We also look at competing trials. Just recently, we were looking at the lung map, which we've had at our sites for some time. And then the Pragmatica study came up and there was overlap on eligibility. So we wanted to assess that with the sites, really dig into it to see what would be a better fit. And I think that you kind of have to make that on a site-by-site basis, what kind of patients that you guys are seeing. But you don't want to we didn't want to just continue to add studies in there and then have 3 or 4 different studies that were competing for our patient enrollments. We also consider the bandwidth, again, Pragmatica S2302, that we realize that there's a 1-hour observation period after the ramucirumab and then a 1-hour observation period after the Pembro. So that's 2 hours of observation, but you have to make sure you have an infusion chair available, the nursing staff available. And for small community centers, that can be a big challenge. So making sure that you have nursing staff, CRCs, who can collect the research samples and send those off at the time that they're supposed to. Lab personnel that's available after 4:30 to spin what you might need. So there's a lot of challenges if you don't have the staffing or bandwidth. Another concern we looked at is patient population. We want to make sure that the patient population supports a consistent guaranteed enrollment, just as Ashley was saying, so that we can meet those requirements. And if you're looking at rare tumor types, that would be a challenge for some locations. And then ultimately, Ashley found this resource and it's fantastic. The SWOG CRA workbench. I know most of us are familiar with it. If you aren't, definitely peruse it. It is a fantastic location to find really great sources. There's a clinical trial review guide that we recently came across. And at UC Davis, within the Cancer Care Network, we're trying to standardize this process because we want to take this form and make it work for us. But it is a really great form. It was developed by the SWOG ORP committee and the protocol coordinators, operations office. And it covers feasibility, protocol development, even just which departments are going to be involved in the clinical trial, which I think we can often overlook that until the trial is actually in process. And then we're going kind of behind the scenes to get everybody else on board and let them know what they need to do. It covers the coverage analysis. My personal favorite, working in regulatory, is regulatory requirements. So we can see that right when you're reviewing a study and not after the fact. And even training, so that you're probably prepared when you're coming into your SIV or site initiation to have everybody on board knowing what they need to do before the study starts. And that's
Awesome. Every time I come to this meeting, almost every time I look at the CRA workbench, there's some new amazing tool or resource that I didn't know existed before. And this, if you haven't seen this one, this form is fantastic. Like Rachel said, this really close communication with our sites is so important during this early feasibility stage. Work really closely with our clinical research coordinators at each site to make sure that we've thought about communication or workflows that we need to establish before we activate this study. Like the best example is S1806 requires that cystoscopy and cytology for the disease assessments. And one of our urologists just wasn't his standard practice to order the cytology at each visit. So we had to really work through that with him and make sure that we had a workflow established. So kind of identifying those potential barriers early in feasibility means that when you are getting ready to operationalize your trial, you're kind of ahead of the game already and already know which folks you need to reach out to and contact. Okay, this is a little off topic with feasibility, but we have two amazing regulatory experts here. So we wanted to ask them the question on how they manage their study teams with regards to upkeeping their delegation of task logs and if they could recommend any best practices to us.
So in regards to the roster upkeep, we are very lucky to have a dedicated rostering individual. Shout out to Nancy, who is in charge of all onboarding and offboarding. She does a wonderful job, kind of like a mini orientation with all of the staff that we onboard. So she's amazing. In regards to DTLs, we have 2 dedicated staff members who are also amazing. Shout out to Kristen and Ron. They keep up with over 500 DTLs because we do everything centrally. So yeah, and then what I can recommend for best practice is communication is key, not only between those 3, but throughout our whole consortium. I mean, it's really important that, our clinical team, for instance, we want to, if a patient is seeing a different mid-level or a different physician, we want to make sure they're on the DTL and rostered with us. As you all know, that's a nightmare later on, about every 3 years. So yeah, and it's also communication is key through all of our sites as well, because I mean they're letting us know who physicians, staff, mid-levels are coming and going. So yeah, communication is key.
Similarly, for our site, the RUMs are it's a literal checkbox on our onboarding and offboarding checklist to make sure we're adding people to all the rosters that we need to add them to. We've got sort of like role specific, which people need RAVE CRA, which people need the lab access, which, I've got that down when I'm adding them to the roster. And I do try to review those at least once a year to make sure that no one slipped through the cracks and I don't have a bloated roster that isn't accurate. And then also we've always designated a backup, and everyone on our team knows who they can go to when I'm not around. So we don't run into any issues where they can't register a patient when they're ready to enroll.
Yes, so I'm really just going to agree with both of you guys on this. Having a central DTL and CTEP ID administrator, I think makes it much easier for the site in general. I'm actually our contact for our site, and I found that we can make those updates in real time. We use the checklist that Ashley just mentioned. Checklists are great if you actually use them. But it really can make it a wonderful transition so that as soon as that person's on board and they've completed their training and they're ready to start that day, I can go in there and update it. And it's not something that could be forgotten. When we did have a decentralized process, I think there was some confusion as to who was doing it and when it was getting done. So appointing that to one person, I would highly recommend, and it did avoid the miscommunication of rules. So I think if it's all done at a comprehensive time and everybody understands that it's done, then I think that can make a big difference.
All right, awesome. Well, that's our show. Thank you so much. I wanted to say it was such a pleasure to work with Anthony and Rachel. Thank you so much for working with me on this. We're all going to be at open forum. Anthony has some pretty amazing tools that he uses for feasibility along with the clinical trial review guide. They'll be at the feasibility table. I'll be leading long-term follow-up. So come see us if you have any questions. Thank you so much.
You turn on the wand. Thank you. This thing is enough to give you a seizure, honestly. Thank you so much, team. And I'd like to introduce our next speaker, who, oh, by the way, a little bit of trivia here. This is Wizard of Oz trivia. Along with Seal Petrowski, Connie Shapanik helped to author that tool that you were just looking at. So if you don't like it, you can tell her too today. Okay. Anyway, darling, I know you're going to talk to us about the most important part, and that's that it takes a lot of heart to do this. It takes our hearts, it takes our patients' hearts, but it takes the involvement of a community. Now, why is that so important? Because it's a voice we have to hear. It's also a voice that the IRB wants to know about. And so I am here to introduce our expert speaker on the subject, Ms. Connie Shapanik.
Thank you, Glenda. So we have such beautiful, elegant science. Our protocols are gorgeous. They're really complex, but if they don't get to people that enroll in them and we can't complete them, they're just dusty tomes on a shelf. So one of the most important parts of this story actually are the involvement and the engagement of our advocates in our communities. We've talked about it throughout how, especially at SWOG, we're really fortunate to have advocates so heavily involved. Advocates, and this is just kind of a summary slide, it's not a didactic, but there's some notes in here if you really want to go home and chew on this a little bit harder and think about some things. There's some notes in here about lessons learned along the way, but they can provide a link to the community, share perspectives and experiences, definitely increase awareness about clinical trials by talking with patients and public that we never get to, help reduce barriers, share experiences, provide resources, act as an advisor, develop materials, just even serve on the IRB and the ethics committee. So our patient advocates often, especially if they've been in one of our clinical trials, seek ways to give back. And this is a priceless treasure nationally, across SWOG and locally. And just before we get to the local story, I just want to talk briefly about our SWOG Patient Advocate Committee and give them a shout out and acknowledge their really important contribution. We have over 25 patient and community advocates here at SWOG. They are deeply involved and are outstanding. So if you've never looked at their resource page on the website, take a look. This is their guide and they are phenomenal. But I want to talk to you a little bit about locally and give you just some things to think about. I know we're all busy and tend to focus on getting the studies done. There's lots of data, there's lots of queries, there's lots of forms, there's lots of everything. But to reach out and have that local connection with our community, one of the things that you can think about is what else do you need in your community? Where can you make a difference? And start somewhere. The story that I'm going to tell you briefly about our group started 15 years ago, and we've done many things, but we didn't do them all at once. So identify a couple of key things that you think would be a real plus, helping your patients in your community move forward, and think about who can help. These two slides are more for your reference later, but you can range everywhere from involving a few patients to formalizing a full-blown community advisory board, which are sometimes complicated. For us, we developed a group of people that started with us in 2008. We were the very first official meeting in our new cancer center that opened. And I was so proud that the first meeting in that building were actually our patients and our advocates. These are some of the things they've worked with me and worked with our board to do. They're instrumental in getting the word out. Things that we can't say, things that we haven't been able to do, places we can't go. They've opened the door for media opportunities, written articles, sometimes even just with a new program at the Cancer Center. They want to know, how does this feel? Our patient advocates have raised their hands and said, well, let's try it from a patient perspective, from the parking lot to the clinic. They've helped us build tools. Our website is a direct output from our patient advocates. They tested it and kicked the tires and said, this is a mess. We don't like this. These words are too hard. Who did you write this for anyway? We don't get it. So we had to start over with our website, but our website is truly an output from our patient input. They've helped us write educational and study materials for other patients to follow. We developed a survivorship care plan communication tool that they specifically developed with and for us. And they also made us a video with $2,000. I mean, who ever heard of such a great thing? And I can't take it off of our website. It's obsolete in a way, but it isn't. It's their story. But helping to build tools that last. They've also been brutally honest. When the financial toxicity protocol first opened, and I thought, oh my gosh, we're going to ask patients for social security numbers. This is going to go really, really well. They were the first ones to say, we don't care about that. You can put our social security number on a bus if it changes things in cancer, so do it. So they gave us really good feedback, and we were actually very involved in that protocol, and that was directly as a result of their input. They gave us a lot of ideas on recruitment strategies. They advised our program leadership. They even helped rename our program. In 2014, when we became an NCORE, we had to change our program name, and they picked our name for us. So they inspire our research team every day. But I just really strongly encourage you, if you haven't directly involved your patients or clinical trials participants in your work, there are lots of little ways, big ways, significant ways. I encourage you, if you need help designing a protocol or asking questions, consider the SWOG Patient Advocate Committee too. They love to hear from ORP. So with that, many hands are on the wheel. Back to Glinda.
Okay, the moment has come. Thank you so much, Connie. Yeah, their crew. Right? We spend all this time getting up to this moment, a lot of work, a lot of assessment, and then we've got to accrue. And so for that, Glinda would like you to look at some of our protocols that are open for accrual right now that could use your time and attention and your patience. S2302 Pragmatica has just opened. This is supposed to be a little bit streamlined, a little bit easier. I love the fact that my personal regulatory person at Davis is nodding her head, which is good news. And I want everybody to kind of slam dunk this one if they can. 1823 already open. SWOG S2010, really great trial. And we all know LungMap. We have lived with LungMap for quite a few years now, and it's been an incredibly successful trial. It keeps adding more cohorts. Please take a look at S1800D and S1900E. There are also a variety of tables that are at open forum, and there are experts in these tables at open forum. So if you're encountering, you know, you have a little comment to make about the specimens and the shipping time or the data or anything else under the sun, they're going to be experts there. So avail yourself of them this afternoon. Deb has gone through a lot of work with all the SWOG folks to make sure these are adequately supported. Excuse me, I have to set my hair. Okay. Do you remember that moment? You know, like the one with the surrender Dorothy thing. God, she is just a problem. Anyway, she kept reappearing throughout all this. And, you know, it would have been easy to just go. And that was the end of that. But, you know, that's not the way the story goes. So we do encounter moments where we can see the Emerald City. Everything looks great. We are so on track. We're about to do it. And then there's a problem. Somebody doesn't meet cycle 1, day 1. Bullet labs, somebody doesn't, you know, can't get there today. They don't know if they can do it anymore because they've had a nervous breakdown at home. We've all gone through all of these things. And so we have brought together some experts today. Amy Kaffernas is a research administrator from Crown Cancer Consortium. Crown Consortium. Jody Cook is a team lead at HSHS St. Vincent Hospital, part of the Crown Consortium. Nicole Mahaffey is an assistant director of PRMS at the UC Davis Comprehensive Cancer Center. And Kristen Ford is the research manager in oncology treatment and quality assurance at UC NCOR. I welcome all of you here to the platform. You're going to tell us how to solve all of our problems. I just know that. And while you're at it, could you work on the witch thing?
Thank you, Glenda.
My pleasure.
All right, so as Glenda kind of alluded, we got the study, you did it, you did the feasibility assessment, you're ready to go, you're ready to get that first patient on, you're all excited. Who's not excited to enroll to another study? And then all of a sudden, you run into a problem. Maybe there was something when you were doing the feasibility you didn't recognize when you looked at the trial that was overlooked, or it was just hidden in the details. We build these feasibilities to do the best that we can, but there is always ends up being a problem. So we brought together a variety of people here from academic community sites, different roles within their programs to help share their perspectives. But one thing I just want you guys to keep in mind, one size doesn't fit all. What's going to work at one program isn't going to work at another, but taking bits and pieces and creating them to be your own really can help with this process and is important. So moving on to our questions. We're going to look at, I'm going to ask Nicole first, when implementing a clinical trial locally, what information from your site's feasibility assessment is utilized to ensure successful enrollment?
Yes, well, our feasibility process has 2 steps. We have a feasibility sign off, which is either yes, it's feasible, but we don't have a way to do this yet, or no, it's not feasible at all. so to implement the trial, we take that process, especially the, yes, it's feasible, but we don't have a process yet, and try diligently for the next 2 to 3 months to invent or create a process with all of our partners to be able to honor the protocol compliantly. So we use that feedback from our feasibility process to do our best to ensure that we have compliant processes set up for a trial before we go live. Our second feasibility check comes from our regulatory department, and it is right before we activate, and it is a mass email to all of our key partners in crime and getting these trials activated saying, are you good? Are we good to go? And everybody gives a final sign off. And this is not so much about the feasibility aspects, but have we solved the feasibility aspects? Do we have kits, right? Do we have drug if we need it on site to start tomorrow? Is everything here? Do we have, all of our coordinators assigned? Was everybody at the SIV? Is everybody trained? Those final checks, and outside departments will respond back to us and give a final approval. Yes, we are good to go. Yes, we are good to go. And then we activate the trial. It's not to say that we don't find things and we have to invent on the fly, but we do rely heavily on our feasibility to inform us of, we can do this as a site. We just don't have a way. You have to put in place a process so that we can work those out before the SIV to train the team on this is what you have to do in order to be compliant with this process.
All right, Jody, how about for you?
Well, at our site, we do a feasibility review. Usually a couple of CRAs will take a look at a protocol and look at it from their perspective on how can we run it, can we open it? And then we also have a regulatory go back and review it too. We do open a lot of studies, like we've heard others on the as needed basis. So we open them up as we know we maybe have a patient. So we're not opening a lot of trials that we're not going to use. But we also look through that feasibility worksheet that they both regulatory and the CRAs look to see if there's concerns, not concerns, or no, we just can't do that. And if there are concerns, are those concerns ones we can overcome? Is it something that, it just needs training, or is it something, no, we don't even have that equipment and we just can't open it here? So we take a good look at that before we open the study. And we also take a look because with us being an NCOR, our site has some satellite clinics, and we take a good close look as it's even feasible at some of our satellite clinics, because some of our satellite clinics can or do radiation and other ones can't. So we've got to take a look to see which ones we can actually accrue to and should open at those sites also. So that's part of the process we go through.
And Kristen.
Yeah, so we really identify that physician champion that can help support and provide buy-in to that protocol that we're trying to open. And along with doing that, we look at multiple other departments to see, are we able to support the trial? Are we able to, really conduct that protocol and follow it to a T? We also look at staff workload. We really assess that. Do we have the staff to be able to successfully accrue to the trial? And then we also look at or work closely with our cancer data management team. They provide us information, tons of information about, do we have the patient population to be successful? And then we also work with other departments and get reviews, and that can take some time, but we just really want to make sure that we're going to be successful and use that feasibility to really try to do a good job.
All right, so everybody's got tools. We all love efficiency. Efficiency is great. Anything we can make easier for our job, the better. So what tools, Judy, do you have at your site to help with the activation and in starting up the process?
And our regulatory has a wonderful worksheet that they use as a checkoff list to make sure that we're getting everything done as we're opening up a study. make sure the details are done, and if a beacon build needs to be done in our Epic system, that they're contacted as soon as we're starting the process to open the study, so that it's up and going by the time the protocol is actually activated. So we're set to go when the patient gets registered. We actually do at our site, a lot of protocols have the oral agents. We actually make our own pill diaries, and we make a drug information sheet. A lot of the protocols don't provide the information to the patients other than in the consent on how to take, when to take, how much to take. So we create a drug information sheet that when the patients pick up the oral agents, it has that information on there so that they can have a quick reference as to when should I be taking this, if I miss it, what I should do, things like that. So that gets all done in the process of us, getting the study open so that we're all set to go when the patient's ready.
Kristen. Yeah, so we use a web-based program called Airtable. It's kind of a hybrid between an Excel spreadsheet and an app. It really helps us to facilitate communication. It helps streamline the process. All staff have access to it. They are able to see where in the process of activating the trial we are, if it's with budget, contract, if it's with regulatory, and it kind of really just helps us keep track of the timeline and how long is it taking us to open the trial? Where do we need to reach out to someone? Do we need to, solve a problem that's been identified? And it also lets us communicate, send messages within that program. So it's been a great, resource. And it also, we use it from startup interest to when the study is completed. So it's really great.
Okay. And we have something vastly similar. We have a clinical trial management system. And I will let you know that we are transitioning it, and it's giving me daily palpitations because all of my reports are going to break, but it's steady state right now is that clinical trial management system does everything for us. It tracks delays. It tracks the steps in our activation process, and it pulls a report that feeds an Excel sheet that auto calculates everything for me. And I track everything with a pending trials team. Our team meets weekly to go through all of these activations and try and discuss implementation issues so that we can get trials activated. And we are vastly lucky that a lot of our outside departmental partners attend that meeting as well. We're only missing one heavy player. And if any of you have buy-in from contracts, come see me, let me know how you did that so that I can get them to come to my meeting. But, and we do track very, very closely utilizing the clinical trial management tool, but we have set up other standard tools as well, just to help the process of the trial. We meet with our outside partners. We have tools that we fill out for every trial for infusion, so that they can track what is needed for them to do, because our infusion nurses are not research nurses, they are clinical staff. We have tools for our IDS partners. We have tools via Epic for our radiology partners when there needs to be a special read on a clinical trial. We have developed a pathway. So we work out standard tools with each department to try and ensure compliance once the protocol is implemented. And it's kind of those tools that we try to develop when we get the, yes, it's feasible, but we don't have a way right now.
Those are definitely some great tips and tricks. Glenda shared some great SWOG studies that have recently opened. And so do you guys have any tips or tricks you'd like to share about a SWOG study you recently worked through that you'd like to share with the members? Kristen.
So this may be pretty simple and self-explanatory, but the most important thing you do, or for our site, is that thorough feasibility review. Knowing that you have the equipment, the staff, the buy-in from your institution is extremely important. You don't want to get all the way to opening and then realize, oh, we don't have that, or no, they're not really interested anymore. It's just a lot of work that's put into these trials. So that is, one of the best tips that I can give. For the recent SWOG S2302, we have launched it at our site, and I can tell you, I read through the protocol. And then I read through it again, and then I looked at the training because I was just like, this is so great. It's so streamlined. It was wonderful. But that kind of protocol really helped us, or helped me to complete the feasibility, complete the reviews, because it was so streamlined. And we were able to get it done. What usually may take a week or two or a few weeks, we were able to really streamline and get it done a lot quicker. So that helped us. But that thorough feasibility review was the best thing we could do.
Cool.
And I would say my tip is highly generic, but it's just communication. When our feasibility reviews come back on federal trials and there's something that is not standard of care, as Anthony said earlier, there's no money, right, to help pay our partners in crime on these trials. So when they come back and they say, we can't do this, it costs our department money, those get escalated to the highest levels of the cancer center, and they have to weigh the importance of the trial against the needs of, people to eat and pay for daycare for their children to keep working on these hard cancer trials. And they come up with a plan to find the funding. And I think that is the hardest part for us with federal trials. That's often the barrier that we have run into. Radiology has recently bent over backwards for us on a trial that ended up costing them a little bit of money. And I think that communicating early, and that's why we run feasibility so early, right, in the process, because it gives you a lot of time to have those conversations and try to just figure out a way. I will say that, like, after I had turned in my slides, so probably just last week, an interesting question came to me from our IDS partners that hasn't even really came up, but I want to share it with you because it stirred an interesting thought process in me. And they're working on a beacon build for an amendment, so it wasn't an activated trial, but they said, would you recommend for this creatinine safety measure that we just build it as if the patient's creatinine is normal at baseline, or should we try to build in the fact that the CT-CTA takes into account abnormal baselines for patients and that their grade 2 would be higher than the normal grade 2, because all the protocol says is grade 2 creatinine can't get treated. And so they're like, is it the patient's grade 2 taking into account their abnormal baseline? Or is it just standard across the board? And the reason why she pinged me is currently I work with the data team at our site as well. And she's like, because if we just do normal, it's not going to line up with the data you input. And then we're going to be holding drug and you're going to say, but the patient's creatinine is normal because it's their baseline. And so it was an interesting question to me. Again, I don't know if this will ever hold up activation. It ultimately went to the PI because they sign off on beacon builds. But it could potentially be something that would hold a build. And I was very excited that CTSU is recommending builds, because then I would say, well, let's see what they did, what they did on theirs. But with that interesting caveat that when the audit comes around, a hold might not line up with an adverse event in that, and there would need to be a comment or an explanation or a tool or a process to handle these types of things. So playing around with that in my head right now.
And Jody, 30 seconds.
Well, once we have a study actually activated, ready to go, we have one of our investigators send out kind of an email blast. And he sends it out and says, hey, we have the study open. It goes to all the providers and even kind of even the ancillary departments that might be involved in the study. So that's really nice. And personally, for like S2010 that we have activated recently at our site, even before we got it open, we were taking a look to see which patients are currently getting chemo or getting radiation that might be eligible for that study once it's open and they're at the point that they're going to be starting their endocrine therapy. So we were kind of watching patients kind of go through the queue and watching when the protocol was going to be open. So we were set for them when it was ready.
Awesome. Well, I want to thank all of our panel members. And thank you guys for paying attention. Nicole and I will be at a table at Open Forums. If you guys have more questions about how to break down these barriers, we'll have some tools to talk about. So looking forward to see everybody then.
And I know that this is absolutely riveting. But I also know that it's time for a bio break. Imagine doing that in the stress. Okay, everybody be back in 10 minutes. Are you there? Cherie, are you there?
Yes, I'm here. Thank you.
Oh. Everyone's still coming in, you know, they don't have enough glitter. They're just not moving. They're coming.
Okay, thank you.
Are we there? Okay. Welcome back to us. All along the way, we've been trying to find wizards. We've been stumbling into helpers, and we have found our friends. And I think it's really important to understand that there's some friends we have yet to meet on this journey. So Amanda Dinsdale is here as the director of the Montana NCORE. Cherie Oxley is the wizard who is calling in from the far, and she will be supporting her teammates. And Barb Lamazny is a lead clinical research nurse at the Cancer Research Consortium of West Michigan. And they are going to speak to us from the standpoint of the dreaded quality assurance. Now, we all know that quality assurance is supposed to be about education and learning to do things better and process improvement and all those wonderful things. And then we get caught with a bunch of queries. And they don't go away real quick. And they're hard to resolve. And they keep coming. And then there's the IPR, which we all know and dearly love because it helps us stay in line. And there have been some wonderful changes we talked about last night where it appears twice a month now. And so we can keep it rolling, right? Always working on improvement. So I'm going to hand off to Amanda, Cherie, and Barb, and they are going to speak to us about site perspectives on quality management, query management, IPRs, and keeping it compliant.
Good morning. We're going to talk a little bit about the challenges that we face in successfully executing a protocol, monitoring and tracking programmatic overview, and maintaining high standards through all the chaos that comes with running trials and collecting valuable data.
So how do we do that? Communication, like Anthony said, communication is key. Communication is going to come in all different forms. At our organization, we send out tons of emails. Our regulatory coordinators send out emails when a protocol has been updated, opened and closed, reconsents. We have a website, our own internal website where things are posted. All these updates are posted. So even if you miss those emails, you can go back to our website and see everything that's been posted there.
Yeah. And this is Cherie, and I agree with Amanda. Communication is key. And one of the things that we do is we send an email out every day at 3 o'clock. And we started doing that just because everyone was so overwhelmed with emails. So it's like a daily post. In that post is any nuances, regulatory updates, go in the post every day. Any kinds of just protocol innuendos that occur. We also let people know what staffing is like. If people, particularly the lead managers, are going on vacation, we keep that information in those posts so that everyone is linked together. We do lots of one-on-one calls with our member institutions, and we have monthly nurses and IRB meetings. So I would agree communication is just crucial. We also have a website that we keep. We actually update it in real time. So that's a resource that everyone knows is available.
One of the other key things with the communication is making sure that you're able to find that information when we get those emails or things that are important updates for a specific protocol. And at our site, when we get those key communications, finding a place where it can be located 5 months later, when you're like, "Ah, I remember there was something that came out." And finding that within your organization, our team has helped us with putting that in a central location for a source of truth to be able to go back. So we're not sending out duplicate questions to study chairs about questions that we have, and being able to just move forward in a swifter way with the correct source of truth.
Yeah, so the key here is help each other. We also do something similar. We have a central shared file where all of these, quirks that we come upon, protocol clarifications that we found, we put them all in a central shared file so we can all look at that, and we're not, recreating the wheel every time we have a new patient for that same study. I always say, if you're in rave or in, looking at your delinquency reports and whatnot, if you see something that's not your work, but it's a colleague within your organization, let your colleague know. Our jobs are hard, you guys, that we have so much work out there. We are responsible for so much. And I feel like we silo ourselves as we're working, but get outside of that. Talk to your peers, talk to your coworkers, let them know, hey, I was in rave, I saw you have a query on one of your charts. It's super helpful. And don't think about it as like nitpicking each other's work or anything like that. It's really helpful. It's a lonely world alone, especially in what we're doing. It's so hard. Work together. I guarantee you, you're going to be, much happier and get a lot more done. And share lessons learned with each other. Just talk with one another. That's why these meetings are so great. Get out there, talk to each other, talk to the people who are next to you. We can learn so much from each other.
I know for our site with the DQP tab and queries, we have one person within the office that monitors those. I don't know if it's a weekly or bi-weekly time point, and then sends those out to our team. And that has been helpful for us as well. And having one or two people that are key in looking at those, especially when you start looking at the ones that are, they break that down into the ones that are within the first, I don't know how many days, but when you get some that are in that 90-day category, we have people that will look in there, find out what it is, reach out to the person who owns that patient, and brainstorm together to be able to resolve the query, and finding out that sometimes we haven't done anything wrong. We are up to date, but it is some glitch within the rave system, and making sure that we communicate that so that we aren't dinged in a report down the road that we were late. We really weren't late. So making sure that you have a system and a team that checks those frequently.
Yeah, and at the Columbus NCORE, so we have clinical managers and leads who respond and look at the queries and address those, and they're divided by research groups. And I think that's been really helpful. Again, it centralizes. We've also been able to tell whether or not there's some issues with how probably a protocol case report form is being interpreted. And also for many of our rural sites, there's one person there. So it helps them not to feel so overwhelmed when a clinical lead or clinical manager is working with them one-on-one to resolve those issues. And if there is a trend that we see, we really use that time also as an opportunity to address also in the monthly nurses meeting, and certainly to do one-on-one educational pivotal moments.
I always say be audit ready at all times. Identify protocol deviations that come up in real time. This helps to address trends before they're repeated. And I know Barb is going to talk more about this in the next slide or so, but yeah, if you can identify those trends in real time, it really does help. We're only audited on a regular audit basis every 3 years. Mm-hmm. So in theory, you could be repeating the same mistake for 3 years. So identify those things up front and a corrective action up front.
One of the things that we have found to be really helpful, that isn't always as practical because of the busy that we have with trying to keep running with all of the patients that we have, is doing a cycle 1 QA. And in theory, it's good if we can pull ourselves off of task to be able to do that. But for every patient that goes through, if you have someone doing a quality assurance check on baseline data and cycle 1 data, we can find where we're misinterpreting what they're asking for in the data and try and catch that early to prevent having 7 or 8 cycles worth of incorrect data entry. So that is something that we are trying to do as a team that's very helpful.
Yeah, and we do that as well. And I think we have, like I said, we have a couple of clinical leads and managers who are assigned by research bases. So they're looking at that data, they're checking rave. And if they find deviations, we really try to address those in real time. And also, I think particularly just a shout out to Debbie Shaw, who is just so important to our program. But one of the things I've learned that she does, which is so helpful, is when she identifies an issue, she really works with the nurse in terms of putting it in the bigger picture so that there's no shaming. No one does usually deviations intentionally, but also really look how it may impact our primary objectives, the secondary objectives. Will the data, we want to make sure that the data that we're collecting, that the volunteer has participated in providing, that data becomes important and or is important, but can be used at the end of the study. We do corrective action plans. Our corrective action plans go to the IRB. They have to be signed off by the treating investigator. They have to be signed off by the PI, clinical manager team. We trend those for any kinds of, obviously, trends that we see. We go by eligibility, toxicities, those kinds of things. And then if we do find trends, you know, that's usually elevated to pull in managers or to work more one-on-one. If we do see trends, usually almost always it's due to new staff or turnover. And we do report, it's been a little bit more fluid with our IRB, but we now are reporting, are providing a summary of deviations to our IRB every 6 months. And if we see trends in that time, we have to report it before, and we also have to report on what we have done to address trends. So it does help us to be audit ready. We also learned the hard way that we really need to do notes to file, because sometimes what may appear as a deviation, there was a lot of reasonings why that occurred, but we might not remember it in 3 years. So we do really, really encourage people to do note to files. Thank you.
Connie, can you advance the slide? So we just want to give a few bullet points of the things that we've talked about here, making sure that we do that quality assurance cycle 1 QA, catch it early, query management, finding the trends, making sure that the data we enter has meaning, and it means it needs to be accurate, taking and learning from our mistakes with educational moments, making sure that we're sharing with everyone within, for us within our consortium, and the protocol clarifications we talked about to prevent duplicate questions, and the real-time CAPAs, making sure, you know, that we know that deviations aren't punitive. It happens. We have missed appointments and scans due to weather, illness, vacation. It's out of our control many times, but tell the story, create that CAPA so that we know ahead of time. And then the QA prep for audits. It takes a village. It takes a village to do that work that we do to make sure that what we're doing is compliant with the protocol. And it takes courage. If you could advance the next slide, Connie. We just want to say to all of our nurses and staff members that we just follow that yellow brick road to best practices, that we take courage. We do have a heart with what we do, and we really do have the brains to do this.
Oh, very much. And thank you, Ms. Japanik, for stepping in. I need to fix my hair and I need to work on the wand. So thank you for covering. So up until now, we have talked about a lot of steps on the journey, a lot of friends, a lot of people involved. But next, we're going to remind ourselves that there is no place like home. Nearing the end of the story, we remember that all of our patients really value, and that's one of the reasons the NCORE program is so successful, being close to home and getting very good quality research. It is a foundation of everything that this endeavor is about, is being able to serve people where they are. So I think it's really important to understand that perspective of someone who goes through this again. We were very fortunate to be able to meet with someone last night. And I understand that Glenda gets to meet someone who is a cancer survivor. So oh my goodness. Hopping right up. Look at her go. Oh. You're a cancer survivor?
I am.
No, you're too young. You're too pretty. You're too everything. Look at that. And look at that. Oh my goodness. I'm going to talk about that. Okay. Well, what kind of cancer did you have?
I had Hodgkin's lymphoma.
My word. And so tell me about that. I don't even know what that is. Ooh.
Well, I don't even know how many years ago. I was diagnosed with Hodgkin's lymphoma while I was in the military, and I had lymph nodes in my neck that were enlarged, and I went through standard chemotherapy.
How'd that go?
Well, it went well.
Yeah,
You know, it's a
right.
fun time. Everybody enjoys that.
Yep.
And 9 months later, I relapsed. Yeah. I don't recommend signing up for it. 9 months later, I relapsed. So 28 years ago, I was sitting at the National Naval Medical Center in Washington, D.C. And my oncologist came in, who happened to have a fellow with him. And they told me that I had relapsed and that there was no treatment for me at that time. But there was a clinical trial available across the street at the National Cancer Institute.
Pause. That's that wonderful moment. That's the same wonderful moment that Toby talked about last night, when serendipitously exactly the right thing happens. And it's because you opened the trial at your site in that one moment. And I would challenge anybody in this room not to tell me that they haven't had at least one of those aha moments in your career, because if you haven't, you will. And in that moment, it's all worthwhile why we do this, because you know it was just the right thing at the right moment. So tell me about it.
Well, I didn't have a clue what that meant. So they said that I could join this, or I potentially was eligible for the clinical trial. So I was ready to pack up all my stuff and walk across the street and start getting therapy. But I didn't know that you had to go through an eligibility check or a consent form, or that it was going to take 28 days for me to be determined if I was going to go on that trial. So I had to go home. I lived in Nebraska at the time. So I went home and they arranged for all of my eligibility check to be done remotely at that time, 28 years ago.
Good job.
And then I trekked to Washington, D.C. and spent 45 days in the hospital undergoing yeah.
Not the Hilton, huh?
No.
Not the Hilton.
No.
Okay.
Mm-mm. So when I checked in, I had two nurses. I don't remember the clinical research coordinators on my case, but I do remember my clinical research nurses. One was Sue and one was Susan, and they played good cop, bad cop. And they told me that every day I was going to get up, I was going to take a shower, and I was going to eat my breakfast, whether I felt like it or not. And then I was going to go through what they fondly called shake and bake.
Woo!
Which was
shake and bake?
It was
Is that
interleukin.
the stuff we put on chicken?
No, it's high dose interleukin 2
Ooh,
in preparation for a
Okay.
bone marrow transplant.
fun times.
Very fun.
You know how to have fun, girl. I want to hang with you. Okay, so then what happened?
So I did my interleukin 2 for 2 weeks of therapy where every morning I got up and I literally shook and baked where my temperature would spike, and yet thought I was freezing. So I was wrapped in blankets. And there's pictures of me and my little head. I'm all bald and I've got my blankets around my head and I'm like, so I did that for 2 weeks, and then they harvested my bone marrow, and then they gave me high dose chemotherapy, and then they gave me my bone marrow back. And this is a very simple way of saying how I went through 45 days. And here I am today. And this is
They
why
gave
we-
you your life back. Yeah. That must have been a really tough experience, but where are you now?
Well, 20 years later, I still work in clinical research. I still do what I do because of the fact that I wouldn't be here today if it wasn't for every single one of you in this room.
So let's remember that this is an individual journey for each and every person, right? How each one of your patients, your research subjects goes through it is different. Every time you're working with each one of them, I think some of the people that are frontline clinical become a little bit chameleon-like, and they match to their patient and they support them the very best that they can. That behind each one of those frontline clinical people are regulatory and data people who make sure that the data is there, the story is told, and the history of the trial, which is really what regulatory is all about, is complete and tells the story in each minute little detail. And none of that was something that Dana knew about at that time. So tell me what you do now.
Well, I am a clinical research manager and oversee a team who do the regulatory, who do the quality assurance, and who see patients through their clinical trials.
How fantastic that you decided to come back and join all the rest of us, much like Toby talked about last night, and give back in a way. Does that offer you a special perspective when you talk to some of your patients?
Absolutely. I always, when I'm interacting with patients or when we're doing any piece of the job, I remember that any misstep, anything that could have deviated on my trial could have impacted the end result. And so I take it very seriously. Deviations are they're something that I really strive to not have.
No.
Absolutely. So it defines every single thing I do at work.
And it makes you very special in giving that time and effort to the patients, just like every one of you are. And that ultimately is one of the messages that we wanted to leave you with today. This needed to become very personal at times, and we'll be talking about that a little bit more after our next speaker. So do me a favor. Would you be off to see the wizard with me? Okay, we'll be handing the mic over to Dr. Michael LeBlanc. Who's going to ensure that we tell the story from beginning to end. And much of what Mike is going to talk to us about is what happens at the Stat Center, how a statistician deals with all the things that we give them, and how we get all the way to publication. Dr. LeBlanc, thank you for joining us today.
Thank you so
And
much.
here we is, if ever
I am reminded it's
if ever
somewhere over the
there
rainbow.
was the Wizard of Oz, is
It's
one because.
definitely a difficult spot to be here after that presentation. I think it's somewhere over the rainbow. So this is a pretty lofty title. It's cure of cancer. It's the huge public health impact. It's all the way to world peace. So it seems like a big title to have, but I'll tell a slightly more modest title, which is just thinking about how that trial comes to fruition. In preparation for today's talk, I try to use artificial intelligence in my job as much as possible. So I asked chatbot, I said, what character in the Wizard of Oz is most like a statistician? And as you can see in the side from this old graphic, it's the Scarecrow. And I only had a brain, right? So apparently the Scarecrow is, he thinks he doesn't have a brain, but of course he does. And so that's why I chose my character. And I've been practicing the lyrics from the song, but I don't think I'll expose you to that today. As Dr. Tangen said very early on, I feel a little uncomfortable, and it's a great thing, but I feel a little uncomfortable in that setting. I'm the faculty statistician for lymphoma and for IMatch, and director of the Statistics and Data Management Center. And I think a little bit of what it's interesting, you're in a statistical sandwich. You started out with Dr. Tangen and you're almost ending up with me. So you went to a lot of good stuff in the middle. So what I'm going to talk about, or what I was supposed to talk about, thinking about the primary analysis. So this is the exciting stuff. This is like, oh, this is the publication. This is the results you see in an ASCO presentation. It might be ways to limit toxicity in some settings. We might be interested in a publication so we can tell our friends about it and tell patients about what happened. It could be an FDA registration for that therapy so we could get it out there forward. And I can't help but being a little bit more transactional, moving a little bit back. Do we just need all that data for the publication? And for those of you who are at CTTC, I'm going to steal a few of my slides from yesterday to say it's not just about the end. It's about the road on the way there, experiences along the road. And so that's going to be the focus of a few minutes that I'm going to talk about today. So this is a result from looks like the font changed a bit there, of a phase 2 randomized study in melanoma, reported out in New England Journal recently, which showed a dramatic improvement in event-free survival for those patients and randomized to neoadjuvant pembro versus adjuvant therapy. And this is the excitement. This is, what happens when you have a well-conducted trial that tests, in this case, an effective regimen change. You see these big differences in survival curves, event-free survival curves. But step back and say, how did we get there? What did we need to get there? And the middle, that was the end, but the middle is, or near the, I would say after listening to the other talks, it's not really the middle. It's a little bit before the end, put it that way. We're monitoring accrual. As we said in other talks, we're worrying about queries and reporting of all the things to the team. The Data and Safety Monitoring Committee, just as this group meeting, is looking at that data every 6 months. So the only way we can get to the end is by having our high quality data and worry about the details that you've all talked about along the way. And more about the middle is, of course, we talked about deviations, IPR reporting. We're interested in all that timely reporting of data. And we evaluated at our end and the study chair's end as well, as we saw from the study chair. Those things are key important steps to get to that high quality end. So, and now I can't resist, since we're talking about every patient's data, and I used these in Oishi a few years ago, but I'm going to take you to there again, because it emphasizes, while I'm while away the hours, thinking about these things, we also have to think about what would happen if in fact we didn't get good quality data. What would happen? What could potentially happen? So this is a thought experiment, an artificial thought experiment, thinking about a randomized clinical trial where there's 2 treatments, and the outcomes of these 2 treatments, the experimental is the orangish yellow line, and the standard therapy is the blue line. And this, if we collected all the data perfectly on this trial, the correct conclusion would be that there's no difference in treatments. But one of the things we really keep an eye on is how patients are being followed, what those data are coming in. And so we said, okay, what if we didn't do a perfect job getting all those data in? And this is that same set of data, that same hypothetical study, if we lost some patients more on one arm than the other arm. And so all of a sudden now it looks promising. That yellow line looks above the blue line, and we might falsely conclude and tell our friends about a therapy that doesn't actually add anything to the story. So it's, again, getting to the end is thinking about the middle and thinking about what the data quality are along the way. And so that's the emphasis that I really wanted to make today. I can't say how important the middle is on the story to the end. So, and then, as I said, the middle also involves us looking at the data and Safety Monitoring Committee looking at the data. And we never know when a study hypothetically will stop for negative results or positive results. And we just, no, nobody can hear me. It's a good thing I didn't start singing there. I would have missed it. So we really just care about all those aspects along the line. And everything depends on the middle to get to the end. But I just wanted to end off with the beginning, because we saw so much of the beginning early on, in the early beginning. None of this works without the protocol, without the operations all the way through, without the sites actually being able to accrue the patients. You can't get to the end, right? And you can't get to the end. So it's everything before over the rainbow is really the important part. And I just wanted to emphasize that. I'm so pleased to be part of this conversation today, and I'm just proud to be part of this group. And anyway, so thank you very much. I'm happy to answer any statistical questions at this point in time.
So, coming home, lessons from the journey. It's been a long journey, right? It is full of twists and turns. It's full of surprises and friends along the way. It's full of, wicked witches and broomsticks and all sorts of ugly things that you got to take care of, right? It is a journey. And that was what we were trying to bring forth to you today. Now, we did add a little bit of humor here, because some messages and some topics go down better with a little bit of humor. But make absolutely no mistake that this is serious business. I know you know that every single day. Okay, I know when we don't have a trial for somebody who has no more options, we all take that very personally. I want you to understand too that there are some key takeaways at this point in time. Some of them are on the slide and some of them I'm going to talk to you about. Among many of the key takeaways, I think courage is one of the biggest ones. We watched Courage in Action last night with Toby. We watched Courage in Action today with Dana. We watch Courage in Action every day that you show up at work and you tackle this again, and you come at it with a renewed sense of spirit of what you are doing, remembering every little tiny piece of this is important to the success of the endeavor. So as our patients and subjects reflect courage to us, we can give that back to them. Don't ever lose that ability. Don't forget the learning lessons that you heard here today. Do the right things, do them in the right order. Don't skip key steps, have enrollment plans, have operational feasibility assessments, take care of long-term follow-up. Those patients matter, okay? Handle the changing expectations. We all know that it's kind of like you get a broadcast and it's sort of a little, what's my next thing? Okay. What's the next priority for me? And in a world where there are many, many competing priorities, it's important to remember that you have to keep at this. Okay. And then remember last that there's a lot of there's remaking ourselves every single day. There's redoing eligibilities, data, a million things like that. There's redesigning, especially during the past 2 to 3 years of a pandemic, a workforce shortage, and really looking at how do we do trials in a different way? How do we decentralize our efforts? How do we make more things possible? And redirecting our resources, our energy, our time. It is all about adapting. It is all about being flexible. It's all about being rigid about the things that are important to be rigid about. But it is all about ultimately resilience. Okay. Never forget that our participants do drive us, and they remind us every single day that we are very lucky. And if we do have cancer, and I tell all of my staff this, if I get it, you'll have me completed eligibility tomorrow. I'll be on the trial the next day, and we'll be successfully treated inside of 6 months, just so you know. So At my age, I can say that because that's a possibility. As we age, our demographic changes and there's a greater chance that we're going to contract cancer. It's a reality of everybody's life. So don't forget that this is tremendously important and it's extremely personal to lots of people. I think everybody in this room has been touched by somebody who has cancer. And so obviously we don't forget. I want to thank you all for your participation in this journey. I hope you enjoyed it. We hoped that you would enjoy it and it would be both informational and inspirational and just worth a little bit of a laugh. And if you want to, there's a burning the dress party immediately following this that you're all welcome to come to. So take care, have a really wonderful, and let me call out Open Forum's the place to be this afternoon. We'll all be hanging around there. There'll be lots of people with a lot more knowledge than I have at those tables. And I hope that you will all take time to come by, see everybody. Remember that Open Forum is like speed dating on steroids, right? You've got 10 to 20 minutes at a table and then you move to another table. If you're not familiar with what table you want to go to, there are QR codes in the middle of each of the tables. And we will see you after the lunch break. Have a great day.